Hi everyone, welcome back to my channel. In this video, I'm going to talk about the concept transcription. Transcription is a process of copying of genetic information from one DNA strand into RNA, or in simple term, it's a synthesis of RNA from DNA. As in the case of DNA replication, even in transcription, the synthesis of RNA takes place purely based on the complementarity. Imagine these are the two DNA strands and this is the RNA strand which is produced during transcription. During transcription, the new nucleotides are added to RNA which are complementary to the nucleotides which is present in this DNA strand. And the DNA strand which is used as a reference strand for the synthesis of RNA is known as template strand. Unlike DNA replication, in transcription, only one DNA strand is used as a template strand for the synthesis of RNA. Why both the strands are not transcribed in transcription? The answer is very simple. Imagine both the DNA strands are used as a template strand for the synthesis of mRNA. In this case, two different mRNA is produced and these two different mRNA will produce two different protein during translation. In such cases, a large quantity of protein will be produced that could complicate the information. On the other hand, these two mRNA which are produced as a result of transcription are complementary to each other, which may bind with each other to form a double-stranded RNA. In such cases, no free mRNA is there to produce protein. So protein will not be produced in that condition. So to avoid all this problem, only one DNA strand is used as a template strand to produce RNA in this case. And that strand which is used as a reference strand for the production of RNA is known as template strand or non-coding strand or antisense strand. And other strand which is not at all used during the transcription is known as coding strand or non-template strand or sense strand. Even in that template strand, only few segments of the template strand will be transcribed into RNA during transcription. The main enzyme that is required for the transcription process is known as RNA polymerase or DNA dependent RNA polymerase. In the case of prokaryote, only one RNA polymerase will carry out whole process of transcription, whereas in the case of eukaryote, it requires three different types of RNA polymerase to carry out the process of transcription. And three types are RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. These three different types of RNA polymerase has the ability to produce different classes of RNA. RNA polymerase 1 will produce rRNA, RNA polymerase 2 will produce hnRNA or precursor mRNA, RNA polymerase 3 has the ability to produce 5S rRNA, tRNA and snRNA. If you study the structure of this RNA polymerase, it's a complex enzyme having mainly two parts, that is sigma factor and core enzyme. Sigma factor and core enzyme together known as holoenzyme. The core enzyme part of the enzyme consisting of mainly 4 subunits, that is 2 alpha, 1 beta and beta dash subunits. And it is this core enzyme part of the holoenzyme or the RNA polymerase actually carry out the polymerization process. And it is a sigma factor which is very important part of this RNA polymerase which helps in the initiation of the transcription. And it initiates the transcription by recognizing and binding of this RNA polymerase to the promoter region of the transcription unit. Now we'll study the meaning of a transcription unit. Transcription unit is a stretch of DNA that codes for RNA, or it is a sequence of the DNA that will be transcribed into RNA. There are mainly three parts of transcription unit, that is promoter, structural gene or RNA coding sequence and terminator. We'll discuss these three parts one by one. First one is promoter. Promoter is a sequence of DNA that is present towards the 5' end of the structural gene or transcription unit. When we are talking about the position of any part of the transcription unit, we are referring to the polarity of the coding strand. So promoter region is present towards the 5' end of the coding strand. As I already told, promoter is nothing but the sequence of DNA of about 6 base long. There are varieties of promoter recognized both in prokaryotes and eukaryote. For example, in the case of prokaryote, the promoter recognized like -10 promoter, -35 promoter, etc. In the case of eukaryote, there are varieties of promoter like Tata box, GC box, etc. As I already told, promoter is nothing but the sequence of DNA. For example, the sequence of -35 promoter is TTGACA. One more important factor about the promoter is it is to this region the RNA polymerase will come and bind at the beginning of transcription. So sigma factor of the RNA polymerase will recognize this promoter region. As a result, the whole RNA polymerase enzyme will come and bind, which initiate the transcription process. So that is all about the promoter region. Second part is structural gene or RNA coding sequence. Structural gene is present between the promoter and the terminator. As the name indicates, structural gene or RNA coding sequence is a sequence of DNA that actually copied into RNA. Third one is terminator region. Terminator region is a sequence of DNA that is present towards the 3' end of the structural gene, or it is present towards the 3' end of the coding strand. This terminator sequence is a sequence that signals the termination to end, and this sequence will stop the termination only after this terminator sequence copied to RNA. Once this terminator sequence copied to RNA strand, it stops the transcription process. So that is about the transcription unit. In the next video, I'm going to talk about the steps of a transcription. Thank you.